introduce our speaker. It's my pleasure to introduce our speaker for today, Dr. Mercedes Pascual. She's coming to us from the University of Chicago, and her PhD in biological oceanography from MIT before doing a postdoc in ecology and evolution at Princeton. She began her tenure track career at the University of Michigan, where she has mentored several different graduate students as well as postdocs, and a lot of them are big names in the field at this point. This includes our very own Katia Cole. And for her career, she has won numerous awards. She became a Howard Hughes medical investigator while at the University of Michigan. She won the Robert H. MacArthur Award for Ecological Society of America, as well as Ecological Society of America Fellowship. And then in 2019, she was elected as a member of the American Academy of the Arts and Sciences. And now in 2015, she moved to the University of Chicago and has been there since. So thank you for coming. Thank you for the
Yes. So, can you hear me well?
Yes.
So yes, it is a special pleasure to be here. In fact, Katia was my first graduate student, and how lucky was that? So that was very fun time. And so, yes, so today I didn't know what to talk about. This IDAS program is the training program. Is I talk about scales and some of the work in climate and infectious diseases that I do relates to that. But I thought I would talk about something different. That is about scales in terms of different levels of organization, and a subject that is very much close to my heart these days and very fascinating. So I like to talk about hyperdiversity in pathogens today, and to basically first mention my main collaborators to previous postdocs who did a lot of good work and are now at their respective positions, Philosoph and Chief. So I said hyperdiversity, and I mean by that fast strain diversity and standing diversity built upon a large pool of genetic variation that involves variant surface antigens. So in a sense, the immune system has to contend with these many phases at once. And although this immune evasion strategy is common in several, if not many, parasites, of not just humans, but livestock and wildlife, it is much less appreciated and understood than the more known brief consequential replacement of a low number of variants in viruses, such as seasonal influenza. And I will be focusing on Plasmodium falciparum, the most virulent parasite of malaria. All of you probably know the life cycle, the complex life cycle. It's vector transmitter, and the sexual phase in the mosquito, asexual phase in humans, mostly in the blood. Now, the high diversity of the antigenic diversity goes hand in hand. Sorry, this typos are. Let me see if I can move the. Can I move this?
You click the three dots under more.
Yeah.
There's a hide floating media control.
Yes, it was. Yeah, it was. That's it. Okay, that's fine. So you can read more or less. Okay, so I said the high diversity goes hand in hand with some very characteristic epidemiological features, and this is for high transmission regions, where we see high prevalence despite high exposure. This would not be possible in a virus such as measles, right? But in our field study site in Ghana, in West Africa, for example, we can sample from 40 to 70% prevalence in the high season, and this is asymptomatic prevalence across ages. Therefore, there is a huge reservoir of transmission for the mosquito to pick up the parasite, and this is an extremely resilient system in high transmission regions. Now, well, sometimes it works, sometimes it doesn't work. Don't ask me why. Okay. So, because I'm a theoretical ecologist, and I'm talking about diversity, I will take a small detour. Basically, there is an enormous amount of theory explaining diversity where it is truly vast. Of course, at the level of organization of species in communities, and it is generally organized in two camps. At one extreme, we have these are peers and then peers again. So let's see. Okay. At the one hand, we have an emphasis on stochastic assembly, that is birth processes of immigration and extinction. In the theory of island biogeography, species come and go, and this elegant extension, the neutral theory of Steve Cabo, where we can understand the patterns of diversity by assuming that species are equivalent. Of course they aren't, but we can assume that those differences do not matter. At the other end, we have a complete emphasis on specific differences because we have an emphasis on specific interactions, for example, competition, mutualism, with resulting niche partitioning limits to similarity. And I would say that it's really going to make it slow. So I would say that the challenge in testing or rejecting neutral theory and understanding which ecological interactions matter, do they matter, is really a problem of identifying patterns, macroscopic patterns, that is large statistical patterns of the ensemble that tell us about the microscopic processes, not all of them, but which ones are dominant. And in doing that, I would say the problem we have had is really which patterns are informative. Here, theory can help us to understand what can be effective signatures to connect data to underlying hypotheses. And returning, of course, to pathogens, the ecological interactions that we are interested in here are the competitive interactions of pathogens for hosts. Because after all, consumer resource systems are, well, infectious diseases are consumer resource systems where we have the infection with the consumer, the resource are the susceptible that do not have immunity, and we all know that the susceptible infected recover dynamics has these typical cycles that reflect this consumption and replenishment of susceptibles. That's in the simple case without diversity. When there is trait variation, the trait variation that matters for this competition is antigenic variation on the surface of pathogens and the specific memory of this antigenic variation. The closer two strains are, in that sense, the more they will compete. And of course, this is specific. And it is frequency dependent because it confers an advantage to being rare, a disadvantage to being common. So the dynamics, this ecological dynamics coupled with evolutionary change has been the focus of now a relatively large body of literature that in fact Katja has pioneered in a sense in the area of phylogenetic, illustrated here with trying to understand the typical replacement of the different clusters, antigenic clusters of seasonal influenza on the right, on different colors. Now, I have this just to point out that the toy molds of this coupling between evolution and competition can give you different outcomes. One can get the typical influenza pattern, and with exactly the same mold, but faster evolutionary, essentially faster evolution with faster mutation, you can get coexisting clusters there. Those clusters should meet, but we cut the tree. And in the mold, you get this coexistence of essentially clades that no longer compete because of limiting similarity. This can be better understood if we now remove evolution and go back to the early molds by Sunetra Gupta of pure competition between a few variants. Consider the simplest example of two loci, each encoding for two antigenic variants, X1, X2, and then Y1, Y2. So a pathogen can take one of these four phenotypes in a sense, and in the coexistence regime, you will find that the types that coexist are those that no longer overlap, And in fact, they are relying on different subpopulation of hosts with different histories of immunity. Therefore, they have generated niches, and the ones that overlap with both are at a disadvantage and therefore are competed. So that's the picture. But what happens if we try to take what we understand at very low dimensions to systems that are incredibly high dimension, where and at two levels of organization, that of repertoires of genes and then the pool of variation that they can construct this variation from. So just to get now concrete, I will be talking about how malaria does this with a multi-gene family that encodes for the major surface antigen in the blood stage of infection. That molecule is a protein. I can attempt to pronounce it. It's Plasmodium falciparum erythrocyte membrane protein 1. Good. So, but anyhow, a genome of a single parasite has 50 to 60 genes in this family. These are known as the VAR genes. Now, they are not linked. They are just drawn this way. They express, when they put inside the red blood cell here, they will export the protein to the surface of this cell where the protein has functions. For example, if the cell becomes sticky, so you have sequestration in the microvasculature, but then it's exposed to the immune system. And what the parasite does is to have different versions of this protein and to basically express them sequentially largely sequentially during infection, which lengthens infection. Now, if one of those types is in memory, your infection becomes shorter because it doesn't get expressed. So that's the aspect of fitness, but you could take some other aspect, it doesn't matter. So you have these multiple types, and you can ask me in a local population at high transmission, how many types are there? So how many, presumably, virgin types or variant surface antigens can we think there are? My colleague Karen Day, a molecular epidemiologist with whom I collaborate, has sequenced part of the gene that is a good marker, and at 96% divergence of the genes, there are of the order of 10,000 or more types. We don't have a genotype phenotype map, but even if not all those differences matter, the combinatorics is enormous. And on top of that, if you can read, the parasite recombines at two levels. Within the mosquito, if you get two different repertoires, they can shuffle genes, and within the blood, the genes themselves can recombine to generate new genes. They can also mutate, but this is a further mechanism of true innovation. So you can throw your hands up and say, well, there is no way there is population structure or anything like strains if on top of this variation, they are recombined. And what I like to do today is to ask whether there is a role for this negative frequency-dependent competition or selection in this intraspecific diversity. So is there strain structure? And are there signatures that tell us that this type of selection really matters in this diversity? And in the second part of the talk, I like to get into this question of diversity at these two levels of organization, because I'm to convince you that it's not by chance that if you are going to play a multi-gene strategy, you are going to play in a very high-dimensional space of high transmission, and that this has implications, in particular for some threshold behavior that may be of relevance to epidemiology. So let me start with an empirical result that I really, really like. Prefer if you could read this. So here we took data from, again, Karen Day, who sampled the blood of all the children in these villages in Gabon. This is from one village, and what you have here You take the blood, you basically sequence as many var genes as you can, and each row is a child with all the types, these types that are recognized at that level I told you, against the same children, the same isolates. And what we are doing is computing for every two children how much overlap there are in the var genes that the parasites carry. So it's a measure of the overlap of the parasites. And you see that this quantity, PTS or pairwise type sharing, all you have to see is that it's very, very low. So that's interesting because almost every child carries different infection or different infections. So that looks very interesting, and then you say, well, it's trivial, right? Because if you have lots of our genes, this may be true at random. Now, we can ask if it is random, so we have randomized this data, and that's the distribution of the mean PTS value over there. The red dot is the mean we observe. Just to say that, okay, there is low overlap because there is high diversity, but there is something, there is a force pushing the system to even lower overlap. So, but of course, non-random doesn't mean non-neutral. Why I say that? Because, of course, we randomize in a computer, but transmission is not a random system. So, I mean, there is a demography of transmission and everything that goes on. So we wanted to ask whether, if we consider that, but we move the specific memory, we would get a different structure than what we have in nature. For this, we actually had to ask, how do we even compare? So we developed some models that under immune selection and neutrality. So the model, I'll tell you in a moment, that has a specific memory is here. What you have seen before determines what you will be, essentially the length of your infection. We have two neutral models, one of complete neutrality, where there is no memory, so the infection has a constant duration. And here, this makes more sense epidemiologically. The duration will be a function of how many infections you have seen before, but not your specific memory. And what we want to know is how to differentiate these. We have a model that is necessarily computational, unfortunately, and we are tracking each individual with its memory. We also have a regional pool of var genes from which we migrate repertoires. So this system is open and then starts to generate variation. We don't have true mosquitoes, but we have some contact rate, and we have transmission. We have, I told you, the recombination during that phase. And another important thing is that each gene has two component blocks, so we can actually innovate by having recombination at the level of the genes. The details do not matter. What is interesting is how did we look at patterns of similarity here? We built some networks in which each node is either a repertoire, in this case a parasite in our model, and the links are a measure of how similar two parasites are. Now that quantity is like PTS, like the genes they share over, except that we did it in two directions because there is something about some priority of, who basically which one goes in the denominator because of the repeating types that can be. It doesn't matter, it's a measure of similarity. Now, of course, this network should be completely connected. It isn't, because we actually threshold it to sort of say, let's look at the structure of the parasites that are the most similar, the ones that should be competing the most strongly. So that's what we did. And just to make a point here, if we start with medium, if we achieve medium var diversity characteristic of places, let's say New Guinea, not yet West Africa, we can actually get the kinds of patterns we expect, and you don't need any analysis to see that these networks are different. These are the neutral ones, and you get these clusters that form under this frequency-dependent selection on these similar things that again are kind of infecting a niche in the subpopulation. But what is interesting is if we go to the high diversity characteristic of West Africa, now you look at the networks and you say, well, it's a bit harder to say how are we going to look at data and differentiate the networks. For example, the degree distribution of the number of connections is the same, and many other things are the same. So Xixin had this idea, it was hers, to basically let a classifier do the job for us and ask which patterns do matter, can we differentiate? So this is a sort of messy slide. The point here is that we basically took an area of parameter space that was reasonable, simulated a lot of networks, and then computed for each set of networks a bunch of network properties that describe distances, degree, motifs, it doesn't matter. And then on the basis of these quantities, we classify the networks to the three regimes, the three sort of types of modes we simulated. The purpose of this is to just show that as a function of two parameters, this is the percent times that we classify correctly, to show that we are classifying correctly. And this is one of the main axes of classification, the amount of variance contributed, just to show in different colors the different properties of the network were contributing. Without going in details, we got this method of classification is method that is used in population genetics to look at structure in genomes, and we use it here for the networks. It's called discriminant analysis of principal components. It basically is using the property of discriminant analysis of looking for groups and the property of principal components of finding major axes of variation. And each point here is a simulation. Therefore, the network classifies somewhere in these two dimensions. And you see the different colors, that basically the different dynamics are, you can recognize them from the networks. And then you can use these to classify data. So this is a small network because we are looking at infections that were essentially had a single parasite, because that's a problem with the data, that you can have multiple parasites and we cannot separate the genes. But what is interesting is that the Ghana data is the dot over there. It basically classifies perfectly with the system under selection. So this is one kind of analysis. We went a little bit further with the networks. Maybe this is not completely necessary because we cannot easily do this with data. But you can look in the model over time at this in this multi-layer network where you have similarity during a season and similarity across seasons. So now you have a network that goes across time, and there is this very nice approach to look at modules in these so-called multi-layer networks. And Shai Philosoph is a master of this, so he had to do this analysis. Here is some information on the software package. But essentially what you will see is in time, the time a module arises and the time it goes extinct. With the idea that the modules may be the closest we have to a strain over time. So what is interesting is here on the top left, you see that modules arise and have a pretty long life in months, compared to the neutral case over here, where there can be modules, but very short-lived. So I found this interesting because one of the hallmarks of frequency-dependent selection, which effectively here becomes balancing selection, is persistence. Persistence, not just in terms of the diversity of the repertoires, but at this case, persistence of modules that are, of things that are similar to each other. And I will tell you later, persistence of the genes themselves. In fact, we know from the study of the genomes that the pieces of the var genes in parasites of humans have similarity to parasites that infect, essentially VAR genes that infect primates. So there is this very long persistence of components. And so what I have told you so far is that the network structure of genetic similarity provides a signature that this competition for host is structuring the parasite population and acting on this diversity. And another signature, again, is this module persistence. So we didn't have the nice modules I showed you from the molds by Sunetra, where you get these very nice niches of too thick, but more than modules, that's not a stochastic mold, but you get these types that cluster. We don't get something so clean, but because we are in a very, very high dimensional space, but we are getting something more messy and more dynamic that still has this signature of competition for hosts. Now, I like to now get to this second part on that emphasizes a little bit the connection to diversity at the lower level. And of course, you can say, well, perhaps it's trivial that if the repertoires get to persist, because there is this, again, this effect of frequency dependence, competition enabling coexistence, then we are going to be large local gene pools. But it's a bit more than that. And I like to basically make the argument that once you start playing with these multi-gene families, at least at high transmission, they are going to be very, very, they are going to be basically, necessarily operating with a very, very large pool of variation. So the idea is here, and it's a fairly simple idea that in fact attributed to Shishin. So basically, we can have sort of a picture where new genes emerge. They're in red at a certain time. They go up in frequency, but maybe they disappear before another gene emerges. In that kind of situation, we may not get a lot of genes accumulated. But if basically the rate of generation is such that it's sort of we get more times generated in the lifetime of the gene, this is of course obvious. We are going to accumulate more diversity in time. But one can write this more. One can write this argument with a threshold quantity we call R diversity, and basically it just has a component which is the rate at which you generate novelty times the mean lifetime of that novelty. Now in the paper, the rate of generating novelty, one can generically write it as the size of essentially the gene population, some sort of mutation or recombination rate, and then the probability that that novelty invades. Here, you get the effect of selection, the effect of how different are you, have you been seen, how many susceptibles you have. The TNU is harder, a lot harder to write analytically. In fact, Shishin did some approximations, but what I want to show you is that the idea is that quantity has to be bigger than 1 to accumulate diversity. And so this is from simulations. If I take 100 years and I let the system evolve, and then from low diversity, and I say that this is the percentage of genes at the end that are new from the beginning to the end. And you see that this is the logarithms. When this quantity crosses 1, regardless of parameters, seasonality, no seasonality, then we have accumulation of new genes when this quantity is larger than 1, numerically. And on average, But what is interesting is that this quantity correlates with a measure in malaria that is a measure of transmission intensity, known as EIR, the number of infected bites per unit time. So basically the higher the intensity of transmission, then that would take you above threshold. The point being that over time, and fairly long times, these evolutionary systems would, at high transmission, accumulate a lot of diversity and build these very large groups. Now, this is seen here over time. Below threshold and above thresholds in the simulations, you see here the accumulation of the different genes in different colors. While here they come and go. In gray, we aggregated all the ones that are at low frequency. Then there are some that come at high frequency, and they come and go, but they are not sticking in the system. So these are very different kinds of dynamics. And the point is that regionally, places like West Africa have the environmental conditions to live here. So they are going to, talking about scales, they are going to have multi-genes with a lot of variation. Now, the argument I would make is that the problem if we start weakening the interactions, so if we reduce transmission, well, the problem or not, depending on whether you want to eliminate the diversity or preserve it, is that, of course, if you weaken the competitive interactions, you are going to weaken also the force that maintains diversity at these two scales. And let me bring it maybe now to epidemiology. So that was over long evolutionary time scales. Now I like to do something to try to connect it to epidemiology. We have some results in the full stochastic simulator, but I wanted to do something that would be simpler and that would be more according to the equations we write in epidemiology for the population dynamics of disease. This is collaborative work with Shichin, but in particular Alfredo Ross. If you don't know him, he is a great theoretician in Amsterdam, the master of structured population models in continuous time and continuous state. So we crossed at SFI, and I thought, well, I need his help because there is no way I'm going to be able to write this model. So indeed, he helped a lot. I'm going to show the idea. So the idea here, if you can see the title, is that where is it? Here. No, then I can go like this, but I'm going to first the previous. So I was interested in whether in epidemiological times we can have a transition between a high diversity system and a low diversity one, because after all, this is what we want to do with intervention. So the idea would be that if you are endemic, you have a lot of diversity, you are resilient to elimination, but over here you are in the more epidemic, low strain and genetic diversity regime that is more amenable to control. And we wanted to see whether this occurs essentially with an important feedback between transmission intensity and diversity generation. So what does that do? And in fact, it doesn't move. The problem is once I get rid of this arrow, then the other thing comes. Okay, so the idea is like this. Is there a positive feedback between the gene diversity in the parasite population, the strain diversity, and therefore the higher transmission intensity, because if you have this diversity, high diversity with low overlap, transmission intensity is higher, and then that if transmission intensity is higher and the population is transmitting faster, you are generating more novelty, and therefore this is a positive feedback. So if you think about positive feedbacks, they can lead to very sharp transitions, and we were interested in that. I apologize for these equations. This is not the full system of equations, but I'm going to use them to explain what's going on here. So we thought, well, maybe we can write some sort of system of equations for this mess. We can and we cannot. So it's a susceptible infected mold, because after all, you are never completely recovered. So it's an SI mold, but we wanted to structure it by memory, not just by age. So when people write these molds, the first equation that says that the derivative of susceptibles relative to time plus the derivative relative to age is standard from age structure population models. But what is not standard is the introduction of this new quantity, which is the fraction of the diversity. And here we are considering the gene diversity, imagining that these are just different variants or antigens. So we don't have a genotype map, but we introduce a new quantity besides age, which is for an individual, how much memory of the diversity that is in the system at that time you have. So this is the number of things you have seen over the total diversity, so this is a fraction. And so this quantity and age characterize now an individual. So we ruled the system with that memory, which means that some key parameters like R, which is the recovery rate, and the force of infection over there, they become a function also of P. There is an equation for P. It takes a lot of going around to sort of sample the 50 genes from D. And by the way, here we are, what we cannot do is to deal with the structure. So I'm sampling at random, and that is the approximation we make. We can then go back to that. So there is, what is important, again, the equation does not matter, but what matters is if I take that system and have a constant diversity, nothing interesting happens. We get the transmission down, the system goes through a standard transition. There were cases, then there are no cases. But if we have a dynamic diversity, so if the total number of genes is changing with the dynamics, then there is a different picture. So we wrote something for that. If you're interested, I can send you this derivation. We wrote an equation where the novelty, of course, comes from recombination, so it matters how many total infections you have in the system. So the more you have infections, the more novelty you are producing. And let me show you what I wanted to show you, which is that this system now has a very interesting transition. It has a so-called tipping point as a critical transition, or alternative steady states. So to look at this, you can look at the fraction infected. So this is your transmission intensity. So for each run of the mole, although this is, well, doesn't matter, this is solved numerically by looking at these branches using algorithms that Andre has. But what is interesting is if you take a particular transmission intensity here, there is one equilibrium up there. So there is a high fraction infected, but there is also a low fraction infected. The low is not zero because we have immigration. And you see they coexist. While here, only the low coexist. So this is an unstable equilibrium. The same with the diversity in the middle. There are two equilibria, and then there is one, and the same with the force of infection. Therefore, the system goes from alternative steady states to crashing to the low equilibrium. So this is interesting because that's what gives you all sorts of interesting transitions, because you can both get stuck down here, which would be good, or get stuck up there. You can jump from one to the other. And so that's more complex. And what is interesting is we actually can compute in the mold the force of infection, something similar to EIR versus the fraction infected for all those points. And then we took the global data set that exists in the literature for the parasite weight as a function of the force of infection. We didn't compare it formally, but there is a suggestion that there is this sharp transition with a lot of noise in the middle. And this is without any calibration. This was just out of the box. But what is interesting is you can say, well, what does it take if I'm up here to get the system to drop? Right? How big a perturbation do I need? Well, we can do something that you cannot do in real intervention. We can separate reducing the number of infections from reducing the diversity. Of course, those you cannot do in practice because one comes with the other. But what this plot shows here is the fraction of diversity that you have to, by how much you have to decrease diversity to go to the lower equilibrium here. And here you go still to the top one. So you see that you have to decrease diversity here at high transmission, high contact rates by about 95% to get the system to flip. While the fractions of infections cured has almost no effect on this. So the effect of the true effect of getting you to this low prevalence is very much influenced by the diversity in this model, and very insensitive to a certain degree to the fraction cure. So this of course has a lot of the existence of alternative steady states has also other effects on control, because you can have an intervention, release it, and under some conditions, if you release it at a slightly longer time, you may stick to the lower equilibrium, while if you release it faster, you may actually rebound very quickly. And that happens because you have this alternative equilibrium. So, as I said, this model is a toy model for now, because after I told you that the selection to keep things apart was important, here we are sampling at random, but once you build a lot of diversity, the overlap is low. So we could add that, but let me conclude. So some thoughts on this. So these hyperdiverse systems may well occur at the opposite end of neutrality, where coexistence is assembled under, as I said, these very specific and frequency-dependent interactions. So the network structure that characterizes similarity of the isolates or infections in this trade space can be useful to basically identify this importance of non-neutral processes. And the negative frequency dependent selection would set the stage for the existence of an unappreciated threshold that concerns the accumulation of the variation on which high biodiversity is built at the next level of organization. So again, we can sort of kid ourselves and mold these systems in low dimension, but for high transmission, that will never work. For epidemiology, I would say that this feedback between transmission intensity and diversity can lead to sharp transitions and tipping points, as we described. And of course, these transitions are relevant for persistence, elimination, depending on whether you are interested in epidemiology or conservation. And here, I should mention that although I've been talking about malaria all the time, as I mentioned, there are other parasites that do this. In protozoa, trypanosoma is well known, and in fact, it has this is of course impossible. They have of a multi-gene family 1,000 copies in a parasite. But there are some bacteria, some fungi. There are also some interesting parasites like I think Anaplasma marginale in livestock. Interestingly, they are all using this kind of structure. And by the way, they are able to have, again, these resilient systems with, never having complete immunity because of the diversity. Now, to close, being an ecologist, I'm talking about scales. This kind of frequency-dependent interactions are very much discussed, for example, in the rainforest as a mechanism for coexistence of different tree species. And here, of course, when we call it density dependence, it's true in the mortality of the trees due to natural enemies. There is evidence of that kind of force. I have it here because I would say that there is a possibility that in these kinds of systems, also the dimensionality of the trade space in which these interactions are playing out may be extremely large, and therefore very hard to test against neutron theory. And I like to end by thanking sort of Karen Day and some people in her lab that have been phenomenal to collaborate. Thank you very much.
Any questions?
That's it.
So for the first part, I have a couple of questions, one on the first part and one on the second part. So for the first part of the talk, you had looked at different kind of network structures and the specific immunity, the generalized immunity, and then the neutral kind of the constant, right? And I'm wondering for generalized immunity and for that constant, I mean, both of those effectively, the generalized immunity affects kind of absolute fitness, right? But it's an effect of relative fitness, right? And so in a way, it's kind of like a neutral model, right?
Yes, it is a neutral model.
So I'm wondering if you compare, like, so the constant one, which you call a neutral model, and the generalized immunity, which is effectively a neutral model, right? So why is the network structure different for those two? The PCA plot.
Yeah, that is a very good question and something that we have to track down the differences, because there is one axis over which they separate.
Yeah.
That is different from the one they separate with the red, right? And it's not, yeah, I haven't yet sort of tracked that down, because the classifier is doing it, and it's actually interesting, like what correlates with that axis versus the other. But there are many properties of the networks, and by eye they look very similar. And so, yeah, it's a very
theoretically you would expect them to look similar, right?
Yes, and yes and no. There is an effect of, it's interesting, right? Although the generalized immunity makes it a lot harder to keep the diversity in the system.
Yeah. Just because of lower absolute.
Yes. So, but otherwise, just by eye, you see those neutral networks look very much like much more how you say tree-like, right, than the others, because nothing is selected. In particular, in the other one, once you have recombination, you are selecting against things that are close to the parents, right? So you start to push things apart. While here you just have so it is very surprising, and I don't know if it is just coming out of the different level of total diversity.
Yeah.
But I think we have to pursue that further. It's interesting, because as with anything where you let the machine do it for you, it's then not necessarily easy to understand it. But that's a good observation.
And then the second question I had was on the second part where you're talking about generation of kind of diversity at the gene pool level. And I'm wondering kind of what you had your N times mu times P big, so basically that sort of, and I kind of missed what the N actually referred as the number of infected individuals. So what so I guess the overall question I have is, what are the assumptions that you're making about how these new genes are actually kind of created? Is it just the number of infections, right, or the number of parasites?
No, here is the number, that is the number of genes.
Okay, so it's actually through duplication.
This is true. This is true. The sort of recombination. Well, it depends. Like that's a generic. The mu there was generic, right? But once we expanded that for a specific system, like Shishin did in the paper, she considered the malaria system, and then she's considering that the mu is mostly this kind of mitotic recombination. So two parents would.
Maybe I missed the mu, it's not generation of new bar gene.
Yes, here it is generation of new bar. That's
so if it's through duplication, though, it doesn't diversity present in the system impact the generation rate also, and then is there like an additional positive feedback which is not in that equation?
Well, there is a positive feedback, but in this equation, yes, there is two things. In the probability of invasion, there is an effect of this diversity on how many susceptibles you have available for the common genes.
Right.
So that is one feedback. But then, of course, what is missing in that equation is the higher level of the epidemiology, right, of the strains. This is although, when you can write in the P invasion, you can write it because when you are writing about the susceptibles available, you have to sort of take into consideration that comes from the overall epidemiology, right? So that was a generic equation, just to make the argument that quantity has to be bigger than 1, so that there is a threshold that is somewhat obvious, although with time, right, in a system that is under this balancing selection, these things should be much closer to 1, right, and be pushed to that threshold, right, in terms of essentially basically saturating the amount of diversity that you can have. But for some low transmission, you're always going to be below. And so the point is simply that if you give it time, you are going to accumulate diversity. Yeah, so that M mu was generic and P invasion was generic. But to write those, you have to then start thinking about the specific system.
Another question. These variants, do you know to what extent the sequence space that they occupy is a constraint? Like what that feeling of diversity is?
Yes, it is. Yeah, and it's a very interesting question because that ceiling is can be constrained by many things, but even in without considering constraints, for example, of the available phenotypes and the genotype phenotype mapping, at some point it's going to saturate as a function of the transmission intensity. In fact, yes, at some point, the genes come in, the repertoires that are coexisting are reaching. There is no other fitness difference here that is different from the frequency. Of course, that's a simplification. In the real system, there are even two subgroups of our genes, one that is more virulent, that is expressed earlier, that we don't bind more with itself. So there is levels and levels of complexity here. But in the simpler case, there is the question of really whether looking at sequencing instead of the real diversity that matters. And that in this system, that is a very difficult question. The genes themselves are very modular, are made of pieces, and the marker we use is just convenient for population studies because it's found in every gene, almost every gene, but the gene is bigger. And recently looking at the sequences and comparing sequences of the full genes, they were able to determine that the relation between that marker and the gene is almost 1-to-1. So it's a good marker, but still that doesn't give a mapping of what is meaningful in terms of what the immune system sees as different. So this threshold has been used traditionally as based on some empirical evidence of different of our genes that are known to be different for the immune system, but there is no general result. Now, even if you take an order of magnitude of this diversity, or even more, the combinatorics are still very, very large. The problem for us, so now we are trying to see if we can fit this kind of transmission model to longitudinal data we have in Ghana through two interventions. So we have the service to the seasons, to the interventions over more than 7 years. And so an exercise we can do is to say, can we pick the model using not just prevalence and other one, but some of the quantities that associate with multiplicity of infection, diversity, overlap, the overlap distribution, and so on. So it's still a question like, for example, could that exercise tell us how much of this variation can be meaningful? So how much of that diversity should we consider as different? I don't know yet the answer, and I'm not sure. So there may be some down the road some bioinformatic approaches to the problem. Certainly, I don't know how else they will sort of get to this math, but they will also start sampling memory in humans in the field. But to do that, you have to select a subset of our genes to begin with to prepare, you know, to do the sort of well, to develop the methods for sampling the memory of humans. So there isn't an easy way.
It's also really interesting to think of how the rate of recombination will depend on how similar they are.
Yes, we have that in the model using a general sort of function that is typically used for this, so that things that are too different from the parents will not be functional. So we have that built into the model. So not everything that we combine is going to succeed here. And we also don't have a good estimate of the rate of mitotic recombination in the field, which is of course very important. It's possible that, so the quantity we have is in vitro, and so we don't know if it applies in the field. So that's an interesting quantity. It's hard to tell because of course if you then allow the system to have an migration of nucleotides, then it's very hard to then separate what is coming from true novel phenotype versus immigration. But clearly, this we are looking at the local population. But in the same way that we can think about building this high diversity over long evolutionary times, we have to imagine that places like West Africa are really a meta population that over a region have this very large diversity. So that also when we intervene, unless it's a regional intervention, you are getting this very high pool of variation available, even if you do not produce novel, right? So it's kind of a very interesting system, but some things we can do in viruses regarding, you know, the epitopes and so on. Of course, we don't have access to here, not for now.
Right.
Yes.
At the great talk, by the way. At the beginning of the talk, you kind of showed like a heat map type thing from the field data, which was like from Ghana, maybe.
On the overlap
Yeah.
of the two of, yes.
And I'm wondering, is there like similar data from areas where malaria is not as prevalent?
Yes,
Like from
this is
because
very
the
interesting.
diversity, yeah.
Very interesting question, and in fact, like, of course, because the transition that is also happening under the hood here, I mean, there is no time to describe all the details, is that when you move from high to low transmission, you are going from monoclonal infections, so essentially infections that are clonal, and each person has just one parasite, and it's just, you know, one kind, one repertoire, let's call it, circulating for a while, right? Versus basically everyone that is infected largely carrying more than one parasite. And so the multiplicity of infection has a nice distribution. It can reach, you know, it's very hard to estimate, and the methods to estimate what they call MOI. So how many different infections did you get, right, if you are infected? That is for most of the people, and certainly the children, is more than one. The median is much higher than one. So, but as you go to low transmission, it is one, right? So that distribution shifts, and so you go from dynamics in low transmission that are essentially a system that is clonal, right, to a system that is doing all this sexual recombination and is getting into multiple infections per individual. And so one thing that is super interesting is that on the practical side of epidemiology is that you can actually use the number of types you have sequenced in an isolate to estimate MOI. So we have done that. Of course, if I tell you, well, the average size is 50 and I sequence 150, you will say roughly you have 3 infections. You had 3 different parasites, right? Of course, you have to consider things about the measurement error in the sequencing and so on. So we have done some of that. So typically, there are some molecular methods to try to get to multiplicity of infection, and people tended to do that with neutral part of the genome with some SNPs, barcodes, and we have been recently we have published a paper comparing that method to actually using the genes under selection. And this for high transmission works better. And then once you have MOI, you can actually compute the force of infection, go from the static quantity to the transmission rate experienced for an individual. But to return, so we can use this part of the genome in a practical sense to do some things that are hard to do in epidemiology at high transmission. But to return to your question, it is really different. And so people have looked, there is a great paper by Shazia Rival in Ecuador looking at VAR genes in a low transmission system. And it's interesting because she can track, there are these monoclonal infections, but there can still be recombination of something that enters in a population. But you can track these different clones coming and going. And this is also why those systems are places where it's much easier to intervene. And not just because there is less prevalence, but because there is less diversity. So yeah, it's a different ballgame. So yes.
So.
Yeah.
So at the very beginning, you said that there is high prevalence, and then also like there are some like asymptomatic patients, and I'm wondering whether like those symptomatic patients have some impact on like selective pressure or like
Yes,
genetic diversity.
yes, they do. In fact, everything I was talking about applies to whether you are symptomatic or asymptomatic. But in fact, the symptomatics almost do not count for the story I told you, because the clinical infections, although they are tragic and they are mostly in the children, they are the tip of the iceberg, right? So really the transmission system and the exposure and the accumulation of memory, once it's not the children in the hospital, it's the children who are getting hit, by repeated exposures, and maybe, they don't end in the hospital, they may have some symptoms or not. Everyone we sample is just, in the village, as they it's not like they are in the clinic or anything like this. So the big, and it's not like a portion, it's really the dominant part of the transmission system is asymptomatic, and it's all, and these people are carrying multiple parasites. So it's remarkable, and somehow of course you are protected. There is a generalized immunity that must be protecting you against certainly that protects you from the severity of disease. This is something we are putting back in the mold, because this mold was used a bit for a piece of theory, but for real epidemiology, we have to have both. And for example, people in previous, in the open malaria mold, which is, for example, an individual-based mold of malaria that is applied in epidemiology, they have generalized immunity building, accumulating over your lifetime, and that determines the intensity of the symptoms and also the transmissibility. So once you get infected by it, how likely it is that you get infected. But the duration of infection seems to be very affected by this kind of specific memory. So we need to do now a better job in our work, at least, to combine the two.
Thank you everyone for coming.